So now we want to consider the biology of viruses. So viruses have been around probably about as long as life's been around, but they were only discovered in the last 100 years or so. And the hypothesis was that there was some material, there's some infectious particle that was actually smaller than bacteria that was causing disease. And so the classic example then was to take sap from, in this case it was a tobacco plant that was infected, and run it through a filter that we knew was fine enough to strain out all the living cells. And then take that sap, apply it to a healthy plant, and then what we saw was that that plant became infected. So there had to be something else that was infecting and causing the disease, but that wasn't alive, that wasn't a cell. So again, viruses are not cells, and they're a small infectious particle consisting of some nucleic acid surrounded by a protein coat. Some viruses, in addition, will have a membranous envelope, so these are the enveloped viruses. And you can see here they come in a variety of different shapes and sizes, and this is just a small example. Now, as we move forward, we need to understand some vocabulary that is associated with viruses. So viral genomes can be double or single-stranded DNA, that makes it a DNA virus, or double or single-stranded RNA. It's an RNA virus. So again, it's unusual to see single-stranded DNA as the storage material. It's unusual to see RNA, either double or single-stranded, as the storage material for this entity. Now, surrounding that nucleic acid, we'll see what's called the capsid, and that is a protein coat. And the individual proteins of the capsid are called capsomeres. Now, in addition to that protein coat around the nucleic acid protecting it, there may be a membrane, and this gets us our enveloped viruses. So that membrane is then derived from the host cell and taken as it's being budded off from the cell that's been infected. Now, bacteriophage is a virus that infects prokaryotes specifically. So phage means to eat, bacteria, the eater of bacteria, but in fact what we know is that it's taking over the host cell and using that host cell to make its own DNA and viral proteins. So again, if we look at the variety, you have viruses that infect plants, you have viruses that infect animals, you have viruses that infect fungi. Basically, if something's alive, it is susceptible to viral infection. This is the influenza virus. You can see here you've got your RNA genome surrounded by the capsids, and this is an enveloped virus, so then you see that membranous envelope also studded then with glycoproteins. So phages are viruses that infect prokaryotes, as we've noted, but in fact they oftentimes have a much more complex structure. And it's really interesting because though the structure looks complex, it is relatively complex compared to the other viruses. It still is a self-assembled structure. Basically, and I'll say this again as we talk about this in more detail, that if you take all these pieces, the head and the tail sheath and the fiber and all these proteins separately and mix them up in a jar, this is what occurs. So it self-assembles. And that's inherent to the properties of the protein, how they're built. So here, this is a great picture. You can actually see the little T4 bacteriophages landing on, infecting, interacting with that prokaryotic cell, that bacterial cell. And that interaction is super important, and it's specific. So every virus needs a cell to reproduce. This is why we don't consider them alive. They need a living cell to accomplish their life cycle. Now, they can't just use any living cell, though. So they have what's called a host range. And this goes back to the idea of protein-protein interaction. Specificity of viral proteins for proteins on the surface of the cell. And only when those interact properly will the virus gain access to the cell. This is why, for example, if your dog has a cold, you don't necessarily catch it, nor do you give your cold to your dog, because the virus is species specific. It has a limited host range. Some viruses have a more expansive host range. They may be able to jump from species to species, and this is going to be incurred when that virus mutates and gets different proteins that now can interact with a different species' cell surface proteins. Now, the basic idea of a viral reproductive cycle is that once the virus gains access to the cell, it's going to hijack the cellular machinery and then use the cellular machinery to make more of the genome, whether that be DNA or RNA, but then express its own genome in RNA and then use the translation machinery in the host cell to make proteins, and then make that assembled virus particle that can then leave the cell to infect other cells.